Hello again. If you've had a chance to watch the first short video on the nucleus, then this should be familiar to you. You should understand what this diagram represents. If you haven't had a chance to watch that video, at the end of this one I'll put a link back to it here so you'll be able to quickly go back and look at it. But let's get on with this topic. This is a nucleus here. And remember, cut in half like that, nuclear pores, an outer membrane and an inner membrane. The nucleoplasm is inside. This line here is to represent the cell membrane. In this video, I want to take a step past just the nucleus itself. The nucleus isn't just sort of floating around in the cytoplasm. It's not just free there. The nucleus, it's actually attached to a whole set of membranes. And that's the topic of this video. This video is all about the endomembranous system, all those internal membranes. So what we have is the nucleus like this, and this gets a little bit awkward to draw, but I'll try and draw it like this. This membrane, I've just put a little space in it there, can be attached to another set of membranes. Now just give me a quick second and I'll try and illustrate this. So you can see what I've got. If I could just take a second and color this space here is inside the membranes around the nucleus. So this is the internal space of the membranes. But notice it's continuous out here and here. So this membranous space between the two membranes of the nucleus continues outside of the nuclear area itself. And as it continues, these flattened membranes actually become a little more irregular in shape and a little wider. I can put more on here like that. I'll just end it there. That's part of the endomembranous system that's inside a cell. And this space that I was coloring purple is continuous throughout this entire system. What this is ER. Endoplasmic reticulum. Endoplasmic reticulum. And the endoplasmic reticulum that's closest to the nucleus is quite flattened out and quite regular, and as the endoplasmic reticulum gets further and further from the nucleus, it becomes more irregularly shaped. These containers are sometimes called cisterns. A little bit small. Simply because they're fluid filled. So they're continuous with each other, but they're fluid filled. So this could be a cistern, that could be another cistern, this could be a cistern over here. And the fluid they're filled with is the same as inside the space between the membranes of the nucleus. There are two types, obviously, two types of this endomembranous system, as it's called. I'll put that term up here. Endomembranous system. Two types of ER in it. The first part here, where it's flattened out, like this, is called RER. The R stands for rough. Where it's here, it's more irregular shaped and actually has a larger volume because it's not as flat. This is called the SER over here. SER. And the S stands for smooth. The distinction in the names, smooth versus rough, is because the rough endoplasmic reticulum has got ribosomes on its surface. And that gives it this sort of roughened, almost sandpapery type appearance if you're to look at it under an electron microscope. The smooth endoplasmic reticulum may have the odd one, but in contrast to rough endoplasmic reticulum, there's virtually none of it. This endomembranous system, there's three domains to this business here. The first domain is the fact that the outer nuclear membrane, this membrane here, that's the first domain. It's the outer nuclear membrane. The second domain is the rough endoplasmic reticulum. And the third domain to this endomembranous system is the smooth endoplasmic reticulum. Now obviously, if you think about it, this system allows for the transport of materials in a cell. There could be something that is in the nucleus that can get into this space by going through a protein or something that's inside this, in the inner nuclear membrane, and get into this space, and then it's just a fluid that it travels through as it may be consumed in another region, and it may move by diffusion because its concentration is greater here. It gets moved along simply because of a concentration difference. We have to look at this a little bit more closely too. The ribosomes on the rough ER, ribosomes, these things are the ribosomes. They make protein. That's actually the location that protein is made. The amino acids are assembled on these ribosome pieces here, and the proteins, as they're assembled, they get put inside the ER. The smooth ER, without all the ribosomes on it, it has a completely different function. What it does is it actually, in this space in here, because it's a greater volume, it's actually enzymatic in here, and this space is where certain lipids get made. So we have phospholipids get made in there, and some fatty acids get made in there. Cholesterol gets made in there if it's a hepatocyte, a liver cell. Liver cells, another function of the smooth ER is they'll actually take things like alcohol and it'll go into the smooth ER, and it'll take the alcohol apart and wind up with a 2-carbon acetate molecule and use the acetate molecule to make fats. So that's one of the reasons, or one of the ways it does that. So it can make lipids by taking ethanol, which is 2 carbons, and it can make convert that into fatty acids. Again, I mentioned hepatocytes a minute ago. If this was a liver cell and it's doing that, you can wind up with people who consume too much alcohol. Their liver cells are working overtime, if you like, converting the alcohol into lipids, and you can get fatty tissue building up in the livers, and that's called cirrhosis of the liver. That's the liver begins to malfunction after a while. So there's the difference between the 3 domains in the endomembranous system. The outer membrane here, which is in communication with the cytoplasm, the rough ER, which is responsible for making ribosomes, and the smooth ER, which actually detoxifies the cytoplasm by taking things like alcohol out of the cytoplasm, and also makes lipids for the use in the cell and even outside of the cell, which is exactly where this topic has to go. These cisterns, whether we're talking about RER or smooth ER, the cisterns can form vesicles. Much the same ways we had endocytosis in another video, where we have the membrane like this, and there's something out here, and it works its way into the membrane surrounded by a vesicle. These things, rough ER and smooth ER, can The process is called blebbing, if you like. If this is the end of a cistern here, it can actually form a little bulge on it. And materials which are being produced in the cistern can be put into here, and it'll break free, and you wind up with a vesicle. Now, depending on whether we're talking about RER or SER, the vesicle could have different contents. And it could have protein in it, obviously, if it's RER we're talking about, or it could have lipids in it, if it was smooth ER we're talking about. Just quickly flip back in your mind to the four major biochemical types that are in cells, in tissues. We have proteins and lipids. We have carbohydrates. Cells are not going to put carbohydrates into vesicles. Carbohydrates, once they're down into the cellular level, are used for generating energy. And the fourth one is the nucleic acids. They're not going to make vesicles of nucleic acids either. The nucleic acid is going to be in here, or it'll be out as RER in, or as RNA in the manufacturing of proteins. So vesicles can contain either proteins or lipids. Now, what happens to these vesicles? Well, I'm going to use an example that I've used several times already. Talk about the beta cells and the islets of Langerhans in the pancreas. These cells make insulin. Insulin is a protein. Those cells have a lot of RER. They don't have much SER because those cells are highly specialized to make a protein, so their apparatus in their cells is designed to make that protein. They've got a lot of RER, and they make vesicles containing a precursor of insulin. Now, what happens to the insulin, or what happens to a protein like that? That's the next step in this story. Let me I'm going to erase this and go to a different location in the cell. If we take, here's our nucleus, I'll just put a circle. Here's our RER and our SER. So this is RER. We're talking about an insulin making cell, so it's going to have that. And it's going to produce vesicles. When insulin is first produced as a protein, it has way more amino acids than it does when it's a functional hormone. It's only got 51 amino acids when it's functioning. Well, when it's produced, it's got way more amino acids than that. So when it's produced, it's not actually functional yet. It has to become functional. But what happens, now that's the same as this, so let me use this space. What happens is this RER produces lots of these. That's what the cell is designed to do. And what happens to these things is they all join together. So they will make what's called, it's still called a cistern. Oops. Cistern, remember, it's just a word that means container. So it'll make a cistern with the precursor for insulin, or if it's another protein, whatever protein it's a precursor for. And this cistern, once it's produced, lines up against other similar cisterns in a set like this. And when you recognize this structure in a cell, this is actually the Golgi apparatus. Golgi is always capitalized. Camillo Golgi was the Italian scientist that discovered the Golgi apparatus. And the Golgi apparatus lies between the nucleus and the cell membrane. So we have a Golgi apparatus. We have the side which the RER, the vesicles, if it was a smooth endoplasmic reticulum, you'd have vesicles with fatty acids in them instead of proteins. So the nature of the topic, or nature of the chemical, isn't important in this discussion. This side here is called the cis face, because it's closest to the nucleus. This side here is called the trans face. So we have a cis face and a trans face of the Golgi apparatus. Now, what the Golgi apparatus does, in a simple sense, is it prepares this product for its eventual secretion from the cell. If we go back and think about beta cells in the islets of Langerhans are constantly making insulin, and they release the insulin into the blood so the insulin can be traveling around the body and do what the hormone insulin is supposed to do. The Golgi apparatus prepares the chemicals for their eventual use. And in this case, in the case of insulin, it's going to be secreted. It doesn't have to be secreted, but in that case, it is secreted. In order to prepare it, there's a number of things have to happen. And there's a separate cisternal location for each of those things. Now, I just have to try and draw your attention back to the topic of metabolic pathways, because what we have is a substance, we'll call it these things here, A's, and they have to be converted into B, which has to be converted into C and D and F and so on. Well, the conversion of A into B is what happens in the first location at the cis side of the Golgi apparatus. So there's enzymes in here. Once it lines up, this is going to get a little bit weird here, but you'll follow it in a second. Once it lines up like this, there's little vesicles of enzymes come out of the adjacent cistern, and that conducts this first reaction. So those enzymes here come out of that reaction, or come out of that cistern, because this cistern doesn't need those enzymes anymore. This cistern now contains this product, and it receives the enzymes from the other one, from the one. So what we have really is, these are little vesicles being donated back. We have a series of chemical reactions, biochemical reactions. Each reaction occurs in a different location. So we've got this series of biochemical reactions, and the first one occurs in the first cistern. The second one occurs in the second location. I didn't say in the second cistern, I said in the second location. I'll get to that in a second. In the third reaction, which is this reaction here, where C gets converted into D, gets converted into that conversion occurs in the third position here. And when I have to stress position, you have to recognize that this process is ongoing, because as these ones form that cistern, there's another set of them being produced. They form another cistern which will line up right here. So we have this cistern's now in the second position, and it becomes in the third position when there's another cistern produced. At the other end of it, we have the final products in the trans cistern here. They've been through all that set of reactions, and this final cistern breaks up into smaller vesicles. And these are the final products. There's a theory, it's called the Cisternal Progression theory, that attempts to explain all of this. What the theory says that is that the cisterns progress, cisternal progression theory, the cisterns progress through the Golgi apparatus. They actually become the next cistern, they become the next cistern. Each cistern has, or each position in the Golgi apparatus has a certain reaction. So if this is, this reaction here occurs in position 1, this reaction occurs in position 2, so this would be position 1, that would be position 2. So as the cisterns are produced, the reactions take place sequentially. A new cistern occurs. The enzymes that were used in the adjacent cistern, that where the reactions already occurred, can be donated back into the incoming cistern. There's two directions of movement here. The cisterns progress this way. This cistern becomes that one, becomes that one, becomes that one, becomes that one, as the reactions are occurring. This is called anterograde movement. Anterograde movement, it's moving forward. The movement of the vesicles this way, because the vesicles that conducted the reaction in this position, after it's conducted the reaction, they're not needed there anymore, so they're donated to the adjacent cistern on the cyst face, on the cyst side, and that reaction can occur in there. This is called retrograde movement, where it's going backwards. Anterograde movement and retrograde movement. It's just a theory. It's believed to be true. So the endomembranous system is a lot more complicated than just ER, because the products of the ER get into the Golgi apparatus, and the Golgi apparatus, in an easy way of saying it, prepares the products for their eventual secretion, if they're going to be secreted, or use in a cell, which is exactly where we have to go in this topic. So there's chemical reactions that occur in here. It's also can be made more concentrated. So there may be a few of them in these, but by the time you get it down here, you put them all together and you get rid of some extra fluid, so they can actually become quite concentrated. Now, these things here, these are vesicles. We've got vesicles up here, we've got vesicles down here. Two different kinds of vesicles. For all intents and purposes, they look the same. These ones up here are called transport vesicles. Because their role, quite simply, is to transport these products from the ER to the Golgi apparatus. They join together, they're transporting that. These ones down here could be secretory vesicles. Because they could fuse with the cell membrane. I'll just erase the CN here. We take this one, for example, and it fuses with the cell membrane like that, and we have exocytosis, and the products are released to the outside. And if this was the beta cells of the islets of Langerhans, this would be the bloodstream. Or if it was in a different type of cell, this could be into the extracellular fluid, or whatever the situation is. But what I said is these could be secretory vesicles. They don't have to be secretory vesicles yet. I drew three of them on purpose, really, because there's three possibilities. They could be secreted just like that. So that's the first possibility. The second possibility is that they could be stored. Storage vesicles. And the case may be neurotransmitters. Your nerve cells, or our nerve cells, make neurotransmitters. A lot of the neurotransmitters are maybe not full proteins, but they're at least amino acid based molecules. So they make neurotransmitters like noradrenaline or acetylcholine, things like that. Or even, if we don't talk about neurotransmitters, we talk about some digestive enzymes. They're made and they're stored in vesicles, ready for their release should they be needed. Right? So they're not automatically dumped out just in case they're needed. No, they're stored should they be needed. So that's the second possibility. The third possibility is that these things here, these particular ones, could be enzymes which are currently inactive, and they're used to make lysosomes. 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 Lysosomes never leave the cell. Lysosomes contain hydrolytic enzymes. When they're active, they contain hydrolytic enzymes which actually are in charge of intracellular digestion. So things in a cell may be worn out or may die, like a mitochondrion might be no longer useful and the cell has to destroy it. So it'll fuse with a lysosome so it gets destroyed. These enzymes that are used in the lysosome, when they're inactive, they're actually marked so that as they come through here, there's an indicator on them. It's a sugar phosphate marker or a tag. So these are tagged with sugar phosphate. As an identifier, and they all wind up in a lysosome. So what happens is this first thing here, the vesicle that gets produced, and it's got the enzyme in it that's got the sugar phosphate tag on it. This thing's actually called an endosome. It's not a lysosome unless it's active, so it's an endosome. And the endosomes are specialized in another way, I suppose is the way to say it, is they have this membrane that they have has protein pumps on it. I'm sorry, hydrogen pumps. Hydrogen pumps. And on their surface they also have that enzyme, there's that that protein we talked about before, clathrin. So that's what happens when these things are made. That's what they look like. So along comes, if we go back to the endocytosis topic from another video, I think I can get rid of this space. I'll just show you what that looks like here. If we have, okay without that, endocytosis occurring, things which are in the environment get into the cell, they're put into a vesicle. This vesicle has clathrin on its surface as well. So clathrin is a protein, it's a labeling protein. Clathrin is a very complex structure. It's like a three-pronged protein that sort of encloses these things. And I always draw them in 2 dimensions, but if I draw it in 3 dimensions, you'd see what it was like. So we've got clathrin around an endosome as well. So there's the clathrin, here's the endosome, there's the proton pumps. Now what happens is these 2 things which are clathrin labeled, they fuse together. So you'll wind up with this sort of thing here. So we have the proton pumps may still be like this. There's the product that got ingested by the cell through endocytosis, and the clathrin is around the surface of it. The enzymes which are in here currently are inactive, right? These enzymes are still labeled with the sugar phosphate label. They're not active yet, but once this fusion occurs, these proton pumps actually start working. They pump protons. Now protons are hydrogen ions, so these things here pump hydrogen ions in. If you pump hydrogen ions in to this endosome, what happens is the sugar phosphate marker gets released, the enzymes become active, and this stuff gets digested. This stuff gets taken apart chemical by chemical and into unit molecules. And from there, after that, these unit molecules can leave and go into the cell. In the process of all of that, these things, which are now an active lysosome, the markers, the sugar phosphate markers, get put in their own little vesicles, and they get shipped back to an earlier stage in the process into the Golgi apparatus for labeling another set of proteins as inactive enzymes. So there you have it. That's a big, long story, this endomembranous system. starting with the nucleus. Think about the nucleus and its connection to the rough endoplasmic reticulum and to the smooth endoplasmic reticulum, and the different functions of those. The fact that you can make proteins for release from cells, and you can make lipids that get released from cells. They all have to go through the Golgi apparatus. We've got that cisternal progression theory, which explains how it is believed that that set of molecules changes sequentially, step by step by step, to result in the production of a vesicle, which is either secreted or stored, or becomes an endosome for lysosome production. Thank you very much. I think that's it.